Today we're going to go over how this genetic engineering stuff is used. How do we use transgenic organisms? How do we use recombinant DNA? How do we use electrophoresis? Everything we eat and much of what we wear comes from living organisms. So genetic engineering is used to try and improve those products we get from plants and animals. The thought is that recombinant DNA transgenic organisms could lead to better, less expensive, and more nutritious food, as well as less harmful manufacturing. Genetically modified plants were introduced in 1996, and they are an important part of our food supply. One example of transgenic plant or a genetically modified plant is bacterial genes are introduced into plants that produce a protein known as Bt toxin. It's harmless to humans and most animals, but enzymes in the digestive systems of insects convert Bt to a form that kills the insects. So insects stay away from them. Plants with the Bt gene don't have to be sprayed with pesticides. In addition, they produce higher yields of crops. Some other genetic modifications of crops include resistance to herbicides, which are chemicals that destroy weeds, and resistance to viruses or viral infections. Scientists are trying to develop transgenic plants that produce food that are resistant to rot and spoilage so that they last longer. They're also trying to develop GM plants that may produce plastics for the manufacturing industry. So the graph here shows herbicide tolerant and insect resistant crops and how we've seen this increase in usage, the number of acres that are planted with genetically modified organisms. Transgenic animals are becoming important. 30% of the milk in U.S. markets comes from cows that have been injected with hormones made by recombinant DNA techniques to increase milk production. Pigs can be genetically modified to produce more lean meat or high levels of omega-3 acids, which are healthy for humans. Using growth hormone genes, transgenic salmon that grow more quickly than wild salmon have been developed. Scientists in Canada combined spider genes into the cells of lactating goats. Goats began to produce that spider silk along with their milk. If the silk is extracted from the milk, it can be woven into a thread that can create a light, tough, and flexible material. Scientists are trying to combine a gene for lysozyme, an antibacterial protein found in human tears and breast milk, into the DNA of goats. So then milk from these goats may help prevent infections in young children who drink it, if their immune system is compromised for some reason. These are genetically modified mice. They have that GFP, that fluorescent protein, in them. Researchers are also looking to cloning to help them make copies of transgenic animals, which would increase the food supply and potentially help save endangered species. In 2008, the government approved the sale of meat and milk from cloned animals. Cloning technology could allow farmers to duplicate the best qualities of prize animals without the time and complications of traditional breeding. Cloning would eliminate that it's less than 100% chance that it would eliminate the mixing and matching. Cloning is not sexual reproduction. There's no different combinations. Golden rice is a genetically modified plant that contains a lot of beta-carotene, provitamin A. This nutrient is essential for human health. 2 genes engineered into the rice genome help the grains produce and accumulate beta-carotene. Beta-carotene deficiencies produce serious medical problems, including infant blindness. So this golden rice, especially in third world countries, it's hoped that it'll help prevent these problems. Other teams of scientists are developing transgenic plants and animals that produce human antibodies to fight disease. Engineers are trying to figure out how transgenic animals could supply us with our own proteins. Human proteins in their milk from transgenic sheep and pigs makes it easy to collect and refine the proteins. These could be used in disease prevention. Transgenic animals can be used as test subjects in medical research. They can simulate human diseases where defective genes are a part of it. Scientists use models based on these simulations to follow progression of diseases and construct tests of new drugs that might be useful for treatment without first testing them on humans. It's been used to develop models for disorders like Alzheimer's disease and arthritis. Recombinant DNA technology can help prolong and save lives. Human growth hormone is used to treat patients suffering from pituitary dwarfism. It's now widely available because it's produced by recombinant bacteria. Insulin to treat diabetes we talked about in class. Blood clotting factors for hemophiliacs come from genetically engineered bacteria. Cancer-fighting molecules such as interleukin-2 and interferon can also be used. Gene therapy is the process of changing a gene to treat a medical disease or disorder. In gene therapy, an absent or faulty gene is replaced by a normal working gene. This allows the body to make the protein or enzyme it needs, which eliminates the cause of the disorder. So here's how gene therapy is supposed to work. So researchers have to engineer a virus that cannot reproduce or cause harm. So they insert, in this case, the normal hemoglobin gene into a genetically engineered virus. They put that into a bone marrow cell and inject that into the bone marrow. So here's the DNA containing the therapeutic gene. It's inserted into the modified virus. The patient's cells are infected with that virus. In theory, I emphasize, in theory, the virus will insert the healthy gene into the target cell and correct the defect. Gene therapy hasn't been fail-safe yet, though. It's actually been risky. In 1999, there was an 18-year-old that volunteered for gene therapy that was supposed to treat a genetic disease that was affecting his liver. He had a massive reaction from the viruses and he died a few days later. For gene therapy to become accepted, we need more reliable ways to insert working genes and ensure the DNA used in therapy does no harm. Genetic testing can be used to determine if 2 prospective parents are carrying the alleles for a genetic disorder like cystic fibrosis or Tay-Sachs disease or sickle cell disease. Genetic tests use labeled DNA probes that can detect the different DNA sequences in the disease-causing alleles. Some genetic tests search for changes in cutting sites of restriction enzymes. Others use PCR to detect differences between links of normal and abnormal alleles. Genetic tests are available for diagnosing hundreds of disorders at this point in time. The same genes are not active in every cell. We talked about that. That's how cells specialize. By studying which genes are active and which aren't, scientists can understand how the cells function normally and what happens when genes don't work as they should. This process is called microarray. It enables scientists to study hundreds or even thousands of genes at once. It's a glass slide or silicon chip to which spots of single-stranded DNA have been tightly attached. Each spot typically contains a different DNA fragment, and differently colored tags are used to label the source of DNA. So if we're comparing the genes in cancer cells to the genes in normal cells, mRNA would be isolated from both types of cells. Enzymes are used to copy that sequence into single-stranded DNA labeled with fluorescent colors. Red for cancer cell, green for normal cell. Both samples would be mixed together, and they would then compete to bind to the complementary DNA sequences already in the microarray. If the cancer cell produces more of a particular form of mRNA, then more red-labeled molecules will bind at the spot for that gene, turning it red. The normal cell produces more mRNA for another gene, the spot will be green. When there's no difference, the spot will be yellow because it contains both colors. The only time DNA is identical is in identical twins because they share the same genome. Chromosomes contain many regions with repeated DNA sequences that don't code for proteins. It's called junk DNA, and these vary from person to person. One sample has 12 repeats between genes A and B, right here. The second has 9 repeats between the same genes. This is from 2 different people. DNA fingerprinting can be used to identify individuals by analyzing these sections of DNA that have little or no function, but that vary widely from one individual to another. Restriction enzymes are used to cut a small sample of DNA into fragments. The repeating fragments of these 2 samples are of 2 different lengths. Then we separate those fragments with gel electrophoresis. They're separated by size. What we're looking for here is a match of the banding patterns. So if you look at suspect 2's bands and the evidence at the crime scene, these match up. These banding patterns correspond to each other. So the DNA fingerprints match. This person, suspect 2's DNA, was found at the crime scene. A DNA probe detects the fragments that have highly variable regions, and that gives us, like I said, the different sized bands. If enough combinations are used, the pattern of bands can be distinguished statistically from any other person in the world. DNA can come from any cell with a nucleus, blood, sperm, or tissue, even from a hair strand if it has tissue from the root. The only exception are red blood cells. Red blood cells don't have a nucleus. The precision and reliability of DNA fingerprinting has revolutionized forensic science, the scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn wrongful conviction. This is actually a low number. Nationwide DNA evidence has saved actually over 300 convicted prisoners from death sentences, 40 some of which are from the state of Illinois. Forensics is used in wildlife conservation as well. African elephants are slaughtered for their tusks, and population has been reduced dramatically. So to stop the ivory trade, African officials are using DNA fingerprinting to identify the herds from which the ivory has been taken. When genes are passed from parent to child, genetic recombination scrambles those markers used for DNA fingerprinting, so ancestry can be difficult to trace. But the Y chromosome never undergoes crossing over, and only males carry it. So Y chromosomes pass directly from father to son with few changes. This helps with paternity testing. Small DNA molecules found in mitochondria are passed from mother to child in the cytoplasm of the egg cell. Mitochondrial DNA is passed directly from mother to child. So your mitochondrial DNA is the same as your mother's, which is the same as her mother's. So it identifies the maternal line. If two people have an exact match, then there's a very good chance that they share a common maternal ancestor. Y chromosome analysis has been used with Thomas Jefferson. Did he father the child of a slave? DNA testing showed that descendants of the son of Sally Hemings, a slave on Jefferson's Virginia estate, carried his Y chromosome. This result suggests that Jefferson was the child's father, although the Thomas Jefferson Foundation continues to challenge that.